How tools made us fat wimps. Responding to posters that read, "Wanted, athletic men to earn $5 per second by holding 85-pound ape's shoulders to the floor," beefy linebacker types would line up at Noel's Ark Gorilla Show, part of a circus that traveled up and down the eastern seaboard of the United States from the 1940s to the 1970s. Inspired to impress the crowds at this star attraction, no man in 30 years ever lasted more than 5 seconds pinning down a juvenile chimpanzee. Moreover, the chimpanzees had to be seriously handicapped, as they wore Silence of the Lambs masks to prevent them from using their preferred weapon, their large canine teeth. Later, the show's apes were forced to wear large gloves because a chimp named Snooky had rammed his thumbs up an opponent's nose, tearing the man's nostrils apart. The organizers of Noel's Ark Gorilla Show were wise to use young chimps because a full-grown male chimpanzee, 150 pounds, is quite capable of breaking a man's back. The authorities did finally put an end to this spectacle, but it wasn't clear whether they were concerned about the young apes or the brawny wrestlers who voluntarily entered the ring with them. How did we become such wimps? It was culture. As cumulative cultural evolution generated increasingly effective tools and weapons like blades, spears, axes, snares, spear throwers, poisons, and clothing, natural selection responded to the changed environment generated by these cultural products by shaping our genes to make us weak. Manufactured from wood, flint, obsidian, bone, antler, and ivory, effective tools and weapons can replace big molars for breaking down seeds or fibrous plants, and big canines, strong muscles, and dense bones for fighting and hunting. To understand this, realize that big brains are energy hogs. Our brains use between a fifth and a quarter of the energy we take in each day, while the brains of other primates use between 8% and 10%. Other mammals use only 3% to 5%. Even worse, unlike muscles, you can't shut down a brain to save energy. It takes almost as much energy to sustain a resting brain as it does an active one. Our cultural knowledge about the natural world, combined with our tools, including our food processing techniques, allowed our ancestors to obtain a high-energy diet with much less time and effort than other species. This was crucial for brain expansion in our lineage. However, since brains need a constant supply of energy, periods of food scarcity, such as those initiated by floods, droughts, injuries, and disease, pose a serious threat to humans. To deal with this threat, natural selection needed to trim our body's energy budget and create a storehouse for times of scarcity. The emergence of tools and weapons allowed natural selection to trade expensive tissues for fat, which is cheaper to maintain and provides an energy storage system crucial for sustaining big brains through periods of scarcity. This is why infants, who devote 85% of their energy to brain building, are so fat. They need the energy buffer to sustain neurological development and optimize cultural learning. So, if you are challenged to wrestle a chimpanzee, I recommend that you decline, and instead suggest a contest based on 1. Threading a needle, a sewing contest, 2. Fastball pitching, or 3. Long-distance running. While natural selection traded strength for fat, increasingly complex tools and techniques for use drove another key genetic change. The human neocortex sends corticospinal connections deeper into the motor neurons, spinal cord, and brainstem than in other mammals. It is the depth of these connections, in part, that facilitates our fine dexterity for learned motor patterns. Recall the plasticity of the neocortex mentioned. In particular, these motor neurons directly innervate our hands, allowing us to thread a needle or throw accurately, as well as our tongues, jaws, and vocal cords, facilitating speech. Reference Chapter 13. Improved motor control was favored once cumulative cultural evolution began delivering more and finer tools. Such tools also created genetic evolutionary pressures that shaped the anatomy of our hands, giving us wider fingertips, more muscular thumbs, and a precision grip. Cultural evolution may also have produced packages for throwing, including techniques, artifacts, wooden spears and throwing clubs, and strategies suitable for using projectiles in hunting, scavenging, raiding, or community policing. The emergence of these, along with the ability to learn to practice throwing by observing others, may have fostered some of the anatomical specializations in our shoulders and wrist, while at the same time explaining why many children are so keenly interested in throwing. More on this in Chapter 15. Alongside these anatomical changes, our species' long history with complex tools has also likely shaped our learning psychology. We are cognitively primed to categorize artifacts, for example, tools and weapons, as separate from all other things in the world, like rocks and animals. Unlike plants, animals, and other non-living things like water, we think about function when we think about artifacts. For example, when young children ask about artifacts, they ask, "What's it for?" or "What does it do?" instead of "What kind is it?" which they first query when seeing a novel plant or animal. This specialized thinking about artifacts, as opposed to thinking about other non-living things, requires, first, that there be some complex artifacts with non-obvious or causally opaque functions in the world that one needs to learn about. Cumulative cultural evolution will readily generate such cognitively opaque artifacts, a point I'll make in spades in Chapter 7. How water containers and tracking made us endurance runners. Traditional hunters throughout the world have shown that we humans can run down antelopes, giraffes, deer, steenboks, zebras, waterbucks, and wildebeests. These pursuits often take 3 or more hours, but eventually the prey animal drops over, either from fatigue or heat exhaustion. With the exception of domesticated horses, which we have artificially selected for endurance, our species' main competition for the mammalian endurance champion comes from some of the social carnivores like African wild dogs, wolves, and hyenas that also engage in persistence hunting and habitually run 6 to 13 miles, 10 to 20 kilometers per day. To beat these species, we only need to turn up the heat, literally, because these carnivores are much more susceptible to warmer temperatures than we are. In the tropics, dogs and hyenas can only hunt at dawn and dusk, when it's cooler. So, if you want to race your dog, plan a 25-kilometer race on a hot summer day. He'll conk out. And the hotter it is, the more you'll beat him by. Chimpanzees aren't even in our league in this domain. Comparisons of human anatomy and physiology with those of other mammals, including both living primates and hominins, our ancestor species and extinct relatives, reveal that natural selection has likely been at work shaping our bodies for serious distance running for over 1 million years. We have a full suite of specialized distance running adaptations from toe to head. Here's a sampling. Our feet, unlike other apes, possess springy arches that store energy and absorb the shock of repeated impacts. This is provided that we learn proper form and avoid landing on our heels. Our comparatively longer legs possess extended spring-like tendons, including the crucial Achilles, that connect to short muscle fibers. This setup generates efficient power and provides us with the ability to increase speed by taking longer, energy-saving strides. Unlike animals built for speed, which possess mostly fast-twitch muscle fibers, frequent distance running can shift the balance in our legs upward from 50% slow-twitch muscle fibers to as high as 80%, yielding much greater aerobic capacity. The joints in our lower body are all reinforced to withstand the stresses of endurance running. To stabilize our trunk while running, our species sports a distinctively enlarged gluteus maximus, along with substantial muscles, the erector spina, that run up our backbone. Coupled with our notably broad shoulders and short forearms, arm swinging creates a compensatory torque that balances us while running. And unlike other primates, the musculature in our upper back allows our head to twist independently from our torso. The nuchal ligament, connecting our heads and shoulders, secures and balances our skulls and brains against running-related shocks. Other running animals also have a nuchal ligament, but other primates do not. Perhaps most impressive of all are our thermoregulatory adaptations. We are certainly the sweatiest species. Mammals must maintain their body temperature in a relatively narrow range from roughly 36 degrees Celsius, 96.8 degrees Fahrenheit, to 38 degrees Celsius, 100 degrees Fahrenheit. The lethal core temperature of most mammals ranges from 42 degrees Celsius, 107.6 degrees Fahrenheit, to 44 degrees Celsius, 111.2 degrees Fahrenheit. Since running can generate a tenfold increase in heating, the inability of most mammals to run long distances arises from their inability to manage this heat buildup. To overcome this adaptive challenge, natural selection favored the 1. Nearly complete loss of hair, 2. Proliferation of eccrine sweat glands, and 3. Emergence of a head cooling system. The idea here is that sweat coats and cools the skin through evaporation, which is fostered by the airflow generated by running. To appreciate what happened, note that sweat glands come in 2 varieties, apocrine and eccrine. At puberty, apocrine glands start producing a viscous pheromone-containing secretion, which is often processed by bacteria to create a strong aroma. These glands are confined to our armpits, nipples, and groin. Guess what they are for? By contrast, eccrine glands, which secrete clear, salty water and some other electrolytes, can be found all over our bodies and are much more numerous on us than on other primates. The highest densities of these glands occur in the scalp and feet, the 2 locations most in need of cooling during running. Measured over body surfaces, no other animal can sweat faster than we do. Moreover, our eccrine glands are smart glands because they contain nerves that may permit centralized control from the brain. In other animals, sweating is controlled locally. It was these innervated eccrine glands, and not the apocrine glands, that proliferated to cover our bodies during human evolution. Because brains are particularly susceptible to overheating, natural selection also engineered a special brain cooling system in our ancestors. This system involves a network of veins that run near the surface of the skull, where they are first cooled by the ample sweat glands on the face and head. Then they flow into the sinus cavities, where they absorb heat from the arteries responsible for transporting blood to the brain. This cooling system may be why humans, unlike so many mammals, can sustain core temperatures above the 44 degrees Celsius, 111.2 degree Fahrenheit limit. At this point, you might be thinking that all these features of our bodies are clearly adaptive. So why would I think that it was cultural evolution that created the conditions that led to the evolution of our species' running adaptations? To get at this point, let's look at three aspects of this adaptive design more closely. First, really putting our endurance abilities into action, where they give us the biggest survival advantage, requires running for hours in the heat of the day in the tropics. When our evaporative cooling system kicks into overdrive, a prime athlete will begin sweating out 1 to 2 liters per hour, with 3 liters being well within our body's capacity. This system can run, and keep us running, for many hours, provided it doesn't run short of a critical ingredient: water. So, where's the genetically evolved water storage system or tank? Horses, which, as I mentioned, can compete with us for distance, do have the ability to store large amounts of water. By contrast, not only are humans unable to consume and store large amounts of water, but we are actually relatively poor hydrators compared to other animals. While a donkey can drink 20 liters in 3 minutes, we top out at 2 liters in 10 minutes. Camels do 100 liters in the same time. How can this crucial element be missing from our thermoregulatory system? Is our otherwise elegant running design fatally flawed? The answer is that cultural evolution supplied water containers and water-finding know-how. Among ethnographically known foraging populations, hunters carry water in gourds, skins, and ostrich eggs. Such containers are used in conjunction with detailed, local, culturally transmitted knowledge about where and how to locate water. In the Kalahari Desert of southern Africa, foragers use ostrich eggs as canteens, water containers, which keep the water refreshingly cool, or occasionally use the stomach sacs of small antelopes. They also use long reed straws to suck water from hollow tree trunks where it collects, and they can readily locate water-bearing roots by spotting certain dry, wispy vines. In Australia, hunter-gatherers created water containers using a technique that involved turning small mammals inside out. Like the Kalahari foragers, they also used surface signs to locate hidden underground water sources. These techniques are non-obvious. Recall that Burke and Wills became trapped along Cooper's Creek for want of such know-how. This reasoning suggests that the evolution of our fancy sweat-based thermoregulatory systems could take off only after cultural evolution generated the know-how for making water containers and locating water sources in diverse environments. The suite of adaptations that make us stunning endurance runners is actually part of a co-evolutionary package into which culture delivered a critical ingredient: water. Supplied with water, any good marathoner probably has the endurance to chase down a zebra, antelope, or steenbok. However, there's more to persistence hunting than endurance. A lot more. Endurance hunters need to be able to recognize specific target prey and then track that specific individual over long distances. Almost any animal we might want to pursue is much faster than we are in a sprint, and will immediately disappear into the distance. To exploit our endurance edge, we need to be able to track a specific individual for several hours by identifying and reading their spoor and anticipating their actions. The ability to distinguish the target, say a zebra, from other zebras is crucial, since many herd animals have a defensive strategy in which they circle back to their herd and try to disappear by blending back into the group. If you can't selectively target the one you've been chasing, the tired one, then you could end up chasing a fresh zebra, a disaster. Thus, persistence hunters must be able to track and identify individuals. Although many species engage in some form of tracking, no other animal tracks the way we do. Studies of tracking among modern hunter-gatherers reveal that it is an arena of intensive cultural knowledge that is acquired through a kind of apprenticeship, as adolescents and young men watch the best hunters in their group interpret and discuss spoor. From the spoor, skilled trackers can deduce an individual's age, sex, physical condition, speed, and fatigue level, as well as the time of day it passed by. Such feats are accomplished, in part, by knowledge of particular species' habits, feeding preferences, social organization, and daily patterns. A number of culturally transmitted tricks further aid persistence hunters. The most interesting of these bits of strategy highlights the subtle, adaptive edge that culture gene coevolution has isolated and exploited. This is a little complicated, so stay with me. Many four-legged animals are saddled with a design disadvantage. Game animals thermoregulate by panting, like a dog. If they need to release more heat, they pant faster. This works fine, unless they are running. When they run, the impact of their forelimbs compresses their chest cavities in a manner that makes breathing during compressions inefficient. This means that, ignoring oxygen and thermoregulation requirements, running quadrupeds should breathe only once per locomotor cycle. But since the need for oxygen goes up linearly with speed, they will be breathing too frequently at some speeds and not frequently enough at other speeds. Consequently, a running quadruped must pick a speed that, 1, demands only 1 breath per cycle, but 2, supplies enough oxygen for his muscle speed demands, lest fatigue set in, and 3, delivers enough panting to prevent a meltdown, heat stroke, which depends on factors unrelated to speed, such as the temperature and breeze. The outcome of these constraints is that quadrupeds have a discrete set of optimal, or preferred, speed settings, like the gears on a stick-shift car, for different styles of locomotion, for example, walking, trotting, and galloping. If they deviate from these preferred settings, they operate less efficiently. Humans lack these restrictions because, 1, our lungs do not compress when we stride, we're bipedal, so 2, our breathing rates can vary independent of our speed, and 3, our thermoregulation is managed by our fancy sweating system, so the need to pant does not constrain our breathing. Because of this, within our range of aerobic running speeds, not sprinting, energy use doesn't vary too much. That means we can change speeds within this range without paying too much of a penalty. As a result, a skilled endurance hunter can strategically vary his speed in order to force his prey to run inefficiently. If his prey picks an initial speed to escape that is just faster than the hunter's, the hunter can speed up. This forces the prey to shift to a much faster speed, which will cause rapid overheating. The animal's only alternative is to run inefficiently, at a slower speed, which will exhaust his muscles more quickly. The consequence is that hunters force their prey into a series of sprints and rests that eventually result in heat stroke. The overheated prey collapses and is easily dispatched. Tarahumara, Paiute, and Navajo hunters report that they then simply strangle the collapsed deer or pronghorn antelope. Persistence hunters can also take advantage of a wide range of other tricks that increase their edge. In the Kalahari, where this aspect has been most studied, hunters tend to pursue game during midday when temperatures are hottest, between 39 degrees Celsius, 102 degrees Fahrenheit, and 42 degrees Celsius, 107.6 degrees Fahrenheit. They adjust their prey choice depending on the seasonally varying health status of their target species, pursuing duiker, steenbok, and gemsbok in the rainy season, and zebra and wildebeest in the dry season. They hunt in the morning after a bright full moon, no clouds, because many species will be tired after remaining active on well-lit nights. When chasing a herd, hunters watch for dropouts, since these will be the weakest members. Non-human predators tend to follow the herd, not the loners, since they rely on scent, not sight and spoor. Perhaps not surprisingly, foragers can spot heat stroke in other people and know how to treat it, as they did with one anthropologist who tried to keep up with the locals, an occupational hazard. Finally, to achieve a running form that maximizes both performance and freedom from injury, humans need to rely on some cultural learning, on top of much individual practice. The evolutionary biologist and anatomist Dan Lieberman has studied long-distance barefoot and minimally shod running in communities around the globe. When he asks runners of all ages how they learned to run, they never say they just knew how. Instead, they often name or point to an older, highly skilled, and more prestigious member of their group or community and say they just watch him and do what he does. We are such a cultural species that we've come to rely on learning from others even to figure out how to run in ways that best harness our anatomical adaptations. Thinking and learning about plants and animals. Over generations, cultural evolution has generated a large and potentially ever-expanding body of knowledge about plants and animals. This knowledge, as we saw with our lost European explorers, is crucial for survival. Given the criticality of this knowledge, we should expect humans to be equipped from a young age with psychological abilities and motivations to acquire, store, organize, extend, via inference, and retransmit this information. In fact, we humans have an impressive folk biological cognitive system for dealing with information about plants and animals. Much research by anthropologists and psychologists, such as the dynamic duo of Scott Atran and Doug Medin, working in diverse human populations, have shown that these cognitive systems have several interesting properties. Children rapidly organize information about plants and animals into 1. Essentialized categories, for example, cobras and penguins, embedded in 2. Hierarchical, tree-like taxonomies that permit inferences using 3. Category-based induction, and 4. Taxonomic inheritance. These are fancy cognitive science terms for rather intuitive ideas. In using essentialized categories, learners implicitly assume that membership in a category, say cats, results from some hidden essence deep inside that all members share. This essence cannot be removed by superficial changes to an individual. For example, suppose you operate on a cat and then paint it so that this individual now looks exactly like a skunk. Is it a cat or a skunk? Or something new, like a scat or kunk? Children and adults will typically say that this is still a cat, which currently looks like a skunk. By contrast, if a table is dismantled and reconstructed as a chair, no one thinks it's still a table. It is what it does. Using category-based induction, learners can readily extend information learned about one particular cat to all cats. If you see Felix go crazy over catnip, you readily infer that all cats will likely similarly respond to catnip. These essentialized categories are assembled over development and cultural evolution into increasingly complex hierarchical taxonomies. With such taxonomies in mind, category-based induction allows people to use their knowledge of one category, say chimpanzees, to make inferences about other categories. The confidence one puts in these inferences depends on the relationship in one's mental taxonomy. For example, knowing a fact about chimpanzees, for example, that they nurse their infants, one can readily infer that wolf moms also probably nurse baby wolves, since both are types of mammals. The tree of relationships also allows us to use taxonomic inheritance. Learners may find out that one of their higher-level categories, like birds, possesses particular traits, such as laying eggs or having hollow bones. When they encounter a new type of bird, say a robin, they can readily infer that it likely lays eggs and has hollow bones, without explicitly learning these facts about robins. Though these patterns of thinking are quite consistent across diverse small-scale societies, it's worth noting that Western, urban populations appear to have some anomalies in their folk biological psychology. In small-scale societies, people typically use, and children first learn, focal categories like robin, wolf, and chimpanzee. But the urban-living children and university students that psychologists typically study use what are called life-form-level categories, such as bird and fish. Moreover, these urbanites seem to preferentially reason from what they know about humans to other species, rather than just positioning humans within the taxonomy and treating them like any other animal. Comparative studies with Maya children, as well as Americans living in rural areas, have suggested why. Urban children receive very little cultural input on plants and animals. Thus, the only critters they know much about are humans. In essence, urbanized Western folk biological systems are malfunctioning due to a poverty of input during cognitive development. This powerful cognitive system organizes the vast body of information that individuals gradually pick up via both cultural transmission and individual experience during their lives. Of course, most of the knowledge that people have about plants and animals comes to them via cultural transmission. To see this system in operation, let's consider how infants respond to unfamiliar plants. Plants are loaded with prickly thorns, noxious oils, stinging nettles, and dangerous toxins, all genetically evolved to prevent animals, like us, from messing with them. Given our species-wide geographic range and diverse use of plants as foods, medicines, and construction materials, we ought to be primed to both learn about plants and avoid their dangers. To explore this idea in the lab, the psychologists Annie Wirtz and Karen Wynn first gave infants who ranged in age from 8 to 18 months an opportunity to touch novel plants, basil and parsley, and artifacts, including both novel objects and common ones, like wooden spoons and small lamps. The results were striking. Regardless of age, many infants flatly refused to touch the plants at all. When they did touch them, they waited substantially longer before doing so than they did with the artifacts. By contrast, even with the novel objects, infants showed none of this reluctance. This suggests that well before 1 year of age, infants can readily distinguish plants from other things and are primed for caution with plants. But how do they get past this conservative predisposition? The answer is that infants keenly watch what other people do with plants, and are only inclined to touch or eat plants that other people have already touched or eaten. In fact, once they get the go-ahead via cultural learning, they are suddenly interested in eating plants. To explore this, Annie and Karen exposed the infants to models who picked fruit from plants, and also picked fruit-like things from an artifact of similar size and shape to the plant. The models put both the fruit and the fruit-like things in their mouths. Next, the infants were given a choice to go for the fruit, picked from the plant, or the fruit-like things picked from the object. More than 75% of the time, the infants went for the fruit, not the fruit-like things, since they'd gotten the go-ahead via cultural learning. As a check, the infants were also exposed to models putting the fruit or fruit-like things behind their ears, not in their mouths. In this case, the infants went for the fruit or fruit-like things in equal measure. It seems that plants are most interesting if you can eat them, but only if you have some cultural learning cues that they aren't toxic. After Annie first told me about her work while I was visiting Yale in 2013, I went home to test it on my 6-month-old son, Josh. Josh seemed very likely to overturn Annie's hard empirical work, since he immediately grasped anything you gave him and put it rapidly in his mouth. Comfortable in his mom's arms, I first offered Josh a novel plastic cube. He delighted in grabbing it and shoving it directly into his mouth without any hesitation. Then I offered him a sprig of arugula. He quickly grabbed it, but then paused, looked with curious uncertainty at it, and then slowly let it fall from his hand while turning to hug his mom. It's worth pointing out how rich the psychology is here. Not only do infants have to recognize that plants are different from objects of similar size, shape, and color, but they also need to create categories for types of plants, like basil and parsley, and distinguish eating from just touching. It does them little good to code their observation of someone eating basil as plants are good to eat, since that might cause them to eat poisonous plants as well as basil. But it also does them little good to narrowly code the observation as that particular sprig of basil is good to eat, since that particular sprig has just been eaten by the person they are watching. This is another content bias in cultural learning. The genetic evolution of our big brains, long childhoods, short colons, small stomachs, tiny teeth, flexible nuchal ligaments, long legs, arched feet, dexterous hands, lightweight bones, and fat-laden bodies, was driven by cumulative cultural evolution, by the growing pool of information available in the minds of other people. Beyond our bodies, culture has also shaped the genetic evolution of our minds and psychology, as we've just discussed, in how people learn about artifacts, animals, and plants. In Chapter 7, we'll examine how eons of adapting to a world full of complex and nuanced cultural adaptations, including tools, practices, and recipes, has led our species to be capable of placing immense faith on cultural information, often trumping our own direct experience or innate intuitions. Later chapters will then explore how cultural evolution shaped the genetic evolution of our status psychology, communicative abilities, and sociality, eventually domesticating us into the only ultra-social mammal. However, before departing on this journey, I want to alleviate any doubts you have that culture can really drive genetic change.